thank you to all of you for joining our seminar on disease modeling in the 21st century, automated organoid assays with 3D imaging. My name is Rob Moody, and I'm an imaging specialist for the United States Southwest, and I'll be your moderator today. Just to let you know, the talk is pre-recorded to try to help mitigate connection issues, but our speakers are both online with us today and be answering questions in the Q&A panel, as well as addressing additional questions live at the end of the talk. So I'd like to introduce our speakers. We have Oksana Sirenko with us from Molecular Devices. Oksana is a senior scientist with Molecular Devices and has been with the company for more than 11 years. She comes to MD via FibroGen, BioSeq, Bayer, and Novartis, and has an immense background in imaging assays and screens, and happens to be my team's go-to person for all our 3D biology questions. So welcome, Oksana. We also have Evan Cromwell, a successful entrepreneur with 30 years of senior executive and technical experience in research, engineering, bioassay development, production, and marketing. So prior to founding Protein Fluidics, he served as director of research at Molecular Devices and was a leader of a cross-OpCo scientific team for the Danaher Life Sciences Group. He was founder and president of Blue Shift Biotechnologies, a high-content screening analysis venture that was acquired by Molecular Devices in 2008. So welcome, Evan. I'm excited for our speakers to show the benefits, ease, and power of automated 3D organoid imaging workflows. And so without further ado, I'll let you guys take it away.
Well, thank you, Robert, for the introduction. I always like this quote from Isaac Newton about standing on the shoulders of giants, because it reminds me as a scientist, I'm really coming up with new discoveries. I'm almost always building upon discoveries of scientists before me of their work. And it's really the case with 3D cell models, where you have very intricate cell models today, organoids that can recapitulate the internal functions of our body, and yet they're built on decades, not centuries of work before us. We think about Russell and Birch in the 50s coming up with the concept of 3Rs, to replace, reduce, and refine. Animal testing with cell-based testings. 2D cell-based models were the first ones. Then in the 60s and 1970, Robert Sutherland published using spheroids, so the first three-dimensional cells grown from cells in a dish, and trying to recapitulate cancer and do cancer drug discovery in improved fashion using 3D models. I would say the most important discovery, though, for current technology, current models, was the stem cells. So being able to take human stem cells and differentiate them to different organ types, and then form them into these complex structures. And then Yamanaka, with his iPSCs, which he invented in the 2000s, really took this to another level where you could do patient-derived, patient-specific organ types and create nearly unlimited number of cells from, let's say, human fibroblasts, if you will. So with these amazing cell models, the government started saying, okay, well, how can we use these? One way was the Tox 21 program, spearheaded by Francis Collins, where you could take different cell models and really focusing on three-dimensional cell models to do safety testing, do ADME tox testing and drug discovery, and then following that was to move into organs on a chip in the tissue chips programs, showing here the beating cardiomyocytes, which are always kind of fun to look at, but these are really, mimicking the heart function in a dish, using patient-derived cell lines. The NIH funded this, the European health authorities have funded this, and most recently, people have been using these to look at COVID-19. And so using organ types to look for viral infectivity, to look the effect of SARS-CoV-2 on the different organ function, et cetera, and really for moving along and moving this discoveries as fast as possible. So today we're going to talk about three different cell model types. We have the single cell spheroids, which I had mentioned, and shown here are HeLa cells. And so the HeLas from the 1950s, which were formed into spheroids back in the 70s, and we're still using them today. Tumoroids, these are from a patient, a breast cancer patient, where the biopsy of the solid tumor has now grown up into a dish to represent their tumor. And then lastly, organoids. So there's definitely some nomenclature here. People refer to a lot of these things all as organoids, but organoids tend to be your multicellular structures that will show organ functionality, sometimes including vascularization, et cetera. So the most complex of the structures. So while their cell models are available, there are a lot of challenges to using them. It takes a long time to grow these up, sometimes weeks, months, even some are years. And once you have them, they tend to be subject to damage. You have to be able to handle them gently. And then how do you do the manual process of media exchange? You're trying to add nutrients or add compounds into these structures. And if you've solved all those problems, you want to know how do you interrogate biology. You want to stain, you want to image them, you want to look for the effects of the compounds on the biology, and all the while maintaining as much of an in vivo environment as you can. One way people have tacked this is using high throughput automation. Shown here is this Scripps High Throughput Molecular Screening Center, and really a nice publication by Tim Spicer and his group down at Scripps, where they did a 1536 well assay using spheroids. It's a single point readout using ATP activity, but people don't normally have access to this type of automation, this type of capability. And if they do, it's often too expensive to use. You really need to have thousands of samples you want to test to make it practical. Researchers have then tended to lab on the chip technologies and microfluidics, which we'll talk about our solution for here. But the promise of microfluidics of lower volumes, smaller footprints, a lot less expensive than the high throughput automation. Those have been realized, and they've been used for single 3D cell model systems. But the chips themselves are often very complicated. You have to hook up a bunch of tubes to them. The chips can be expensive. You're using PDMS, it's a very manual process to put these together. And they tend to be single purpose. So if you have a wide variety of experiments, you have to use different types of chip for different types of setups for those experiments. So we looked at what researchers want, and it's sort of a cliche, but they want something easy to use that they can use for a variety of different organoid types, and be able to automate a lot of the steps to take out the manual interventions or improve variability, et cetera. And lastly, to interrogate the biology, these have to be compatible with the multimodal downstream analysis. So we have worked with molecular devices to combine our microfluidic automated organoid system with their high content 3D imaging systems to really produce something that can help you do your 3D cell model assays. And we believe that this can have a big impact on your ability to do drug discovery, biological research with these exciting new cell models. So let me introduce you to the PUMA system. PUMA is a pneumatic microfluidic assay system doing automated organoid assays. The system fits in an incubator, so you can run your assays over hours or days, if you will, in a true in vivo environment. Because the processing is done inside the environment, you can do what we call in situ sampling. And it's a hands-off workflow. So once you've prepared your plate and put it in the system, it runs automatically, preparing all your samples for your downstream analysis. We mentioned wanting an ease of use. So we've kept the workflow as simple as possible and fit right in with your current laboratory workflow. The plates and chips are loaded with your organoids and reagents, and there's templates for loading those to tell you where to put the different reagents, and I'll show some examples of that later. Once the organoids are loaded, that goes into the system. The interface between the PUMA system and your samples is really a simple clamshell operation. So you seal the pneumatic system to your flow chips in a very simple one-step operation. The assays themselves are run by protocols, which are preloaded on the PUMA system. Simply press play, and your assay starts running inside your incubator. So you can see here it fits comfortably inside standard incubators. There is gas exchange between the outside of the PUMA system and your flow chip, your media. So you get the exchange of CO2, et cetera, to your media. And then once the assay is finished processing, the plate comes out, and then you can put it into your high content imaging system, your plate reader, or whatever other device you have for your downstream analysis. I mentioned the protocols. So these are preloaded protocols. They determine the steps, assay steps that you're going to run. You simply select the protocol you want with the incubation time that's predetermined, and then press play. And so then that runs the assay, and then there's a touchscreen driven interface here, which will give you information on time remaining, where the system is in the process, and when the system is ready for you to unload your plate. There's a offline program that you can then use to set up and modify or customize these different protocols. And you can see the USB port here, which is made to load protocols onto the system. So if you want to run a 72-hour protocol, you can modify this, add additional time, load the protocol onto your system in the incubator, and off you are, ready to run. At the heart of the system is our 3D Flowchips. Now this looks a lot like a 384 well plate with a whole bunch of wells here, but it really is a set of 4 independent Flowchips, and each Flowchip has 8 lanes, each lane being 1 test. So you have 8 tests in 1 Flowchip, and then 32 tests total in the holder here. The holder conforms to the SLAS ANSI standards, and so it's compatible with any automation, plate handlers, liquid dispense, plate readers, imaging systems, etc. And then you can also put a lid on it so you can keep it sterile or keep humidity, keep evaporation down, etc. So it's, again, compatible with all laboratory automation that you currently would have. If you look at the side view here, you can see that these wells are really reservoirs connected by microfluidic channels on the bottom. So each well is about 20 microliters. The middle one here, with the larger field of view, is where your sample would be placed. And then you have connecting reservoirs, which you can then load different reagents, whether it be your media you want to exchange, compounds, staining solutions, etc. So any of the assay reagents you normally use be loaded in here and then run automatically. At the heart of our system, one of the keys to our technology is what we call our protected sample chamber. So when the spheroid or organoid is loaded into the flow chip, it moves down into the bottom, and it's in this chamber down here. Now this is about a 1.2 millimeter diameter chamber, around a 1 microliter of volume. So as you're doing exchanges, there's 20 microliters in here, so about 5% of your fluid is down here. And what that enables is, as you're doing transfers out of the well or transfers into the well, your organoid stays within the media, so it doesn't dry out, it's not perturbed by the flows, it stays in the middle for your imaging, and so minimum perturbation to your organoids during these fluid exchanges, as opposed to having a pipette tip come down here, which might suck up your organoid, or the pipette tip might hit it to damage it. So it's a very elegant way of allowing you to do reagent exchanges in an automated fashion. Now, I should say that there's 7 ports that come in from the side. We also have a port that comes from the bottom. It's not shown here. So if you do need to drain all the fluid out of the well, you can do that. So if you're worried about any residual media or compound before a next step, if you will, you can drain the full well of the media or the compound. The second animation here shows how we do the fluid transfer steps. And so with the organoid in the protected chamber here, you can then drain fluid out, you can transfer in fresh medium, and then as the system is processing, it will sit here and incubate. As I mentioned, there's gas exchange going from the incubator environment to the media here, so you can keep the media happy, if you will. And at the end of the incubation period, then you can remove that media, move in extra media, or you can do compound dosing. Again, have a predetermined incubation time for that step. At the end of that, then you can transfer that back out. And here you can now store this for collection, or if you want to do downstream analysis for secreted factors, cytokines, growth factors, etc., or metabolites. Your media is now collected here. And that can be done in multiple steps. So you could do multiple media transfers and collect a time course of supernatants if you wanted to, all automatically. As this runs again, we show the media transfers occurring. And then after the transfers are done, you now can prepare it for sort of downstream analysis, where you then, for example, move in a staining solution. The staining solution can then sit in the well for a predetermined amount of time. You can remove the sample plate at this point to do imaging. If the stain produces higher backgrounds and you wanted to do that, you could drain the staining solution and move in a wash buffer. I mentioned being able to remove the samples to do imaging. Because of the way the system is set up in the protocols, at any point in your protocol, you can stop the system, remove the plate, inspect the organoids if you want to look for phenotypic changes that are occurring during the course of your assay, or if you wanted to sample supernatants during the course of the assay, you can remove the plate and then simply put it back in the Puma system and recontinue your assay protocol. He shows examples of the imaging, and a number of microfluidic devices, you have trouble imaging through them just because they have PDMS or they have other plastics which are really not amenable to good optical transmission. In this case, we have 10 COC bottom sealing art chips, so you can do very high resolution imaging. This is the 20x water immersion lens. Oksana will show some other examples of this later, looking at some islets and looking at some lung organoids doing confocal imaging stack. And so the ability to do this imaging, I think, is a clear advantage of our flow chip design. Let me show you an example of assay workflow. And this we adapted for a researcher who is doing diabetes work. And he was, in his current workflow, he looks at islets. These are harvested from mice. And he wants to look at for secretion of insulin and glucagon from the islets and look at the factors that affect that secretion. His current workflow, he takes 7 to 10 islets and puts them into about 200 microliters of a glucose solution. He then incubates those in a water bath for 16 minutes and then removes the sample for downstream analysis, for immunoassay analysis. So for him, he's getting 1 data point per 7 to 10 islets. So the workflow we came up with is allowing him to use less islets and to collect more data points per set of islets. And so here, the islets and glucose solutions are loaded into our flow chip. In the first step, a low glucose solution is transferred in and it's incubated. Then that gets transferred back into well for collection after the assay is over. We then re-equilibrate the islets with normal glucose levels and then transfer in the high glucose solution to look at the effect of a higher sugar level, higher glucose levels on the insulin secretion. And then that gets transferred back. And the last step is to lyse the islets and do that to collect the total insulin or glucagon levels in the islets, a normalization step. These are all done automatically. So at the end of his process, he's now generated 3 samples, a low glucose solution, a high glucose solution, as well as the lysed islets for normalization. You could also throw in a 3rd or a 4th step here if you wanted to, if you want to look for effect of different compounds on the insulin secretion, you could do that. So from the reduce, refine, replace mantra, he's now reduced the number of islets he's using from say 7 to 10 down to 3, and he's collected twice as many data points. You can collect 2 or 3 as many data points from each set of islets. So that not only reduces the number of mice he has to use, which is expensive, but it also improves the reproducibility because now you're collecting the same information from the same set of islets as opposed to having the average cohorts of islets together. This just shows an example of one set where he ran islets looking at the 1 millimolar, 11 millimolar secretion levels, seeing insulin secretion go up as expected with good statistical separation between the two groups. A second application is in the metabolomics. So this is researchers studying cancer, and looks at the metabolism and how it relates to the cancer growth and cancer propagation. For this particular case, he was very interested in looking at metabolism and capturing it in situ. You know, the current workflow, you would remove the cancer spheroids, from an incubator, then put them on ice, and then ice you have to then wash them and remove the media and then lyse them colds. So it's really an unknown perturbation to biology from removing them from the incubator, and metabolism can be very sensitive to these, especially over the short term, the 3 to 5 minute period from when you remove them from the incubator and then get them frozen on ice, if you will. So in the Puma system, he's now able to move the lysing solution into the organoid chamber in the incubator, so no perturbation to the temperature or the CO2 levels there, and then collect that lysate sample and run that for metabolomics analysis. So the ability to in situ lysing is key, and then the ability to see the high signal to noise from single spheroids is also very important. Because we're lysing into 20 microliters, there's a natural concentration effect of the metabolites. And it works well with the LC mass spec, which uses about 10 microliters a sample. And so this is showing metabolites that we analyzed from a single colon cancer spheroid, around 2,000 cells. And we're able to see around 240 metabolites with very good signal to noise from that. And then you can show the effect of different treatments. Here we did star-sporin's treatment and a C59, which is a Wnt pathway inhibitor treatment. And you see nice changes in the metabolomic profile of the spheroids based on these different treatments. And so this is a very exciting area that he's using to look at melanoma cancer, and we expect some very nice results coming out of that in the future. So just to wrap up this portion, I've introduced you to the Puma system for automated organoid assays. Allows you to incubate your organoids, spheroids, tumoroids in different media, perform media exchanges, compound additions, allows you to do in situ sampling of supernatants, in situ lysing to capture metabolomic profiles, or to do normalization to total cell number of your organoids. And the plate format allows it to be ready for analysis. And so your organoids are held in a system that's compatible with high content imaging. It has a optically clear bottom, thin COC bottom that allows you to do high resolution imaging. It also allows you to do fluorescent readouts, luminescence readouts, and other types of downstream analysis, which I haven't shown here, but we have application notes on our website if you would like to see that. So with that, I'll now turn it over to Oksana to show uses of the Puma system for doing organoid assays using 3D imaging.
I'm Oksana Sirenko. I'm a senior scientist at Molecular Devices, and I will be talking about 3D imaging and analysis of complex biological systems. Biology nowadays trends toward increased complexity of assays. It includes 3D microtissues, organoids, organ on a chip, which represent transition between cell assay to tissues and organs. Then technologies and instruments need to increase sophistication and complexity and capabilities to enable scientists to work with and automate those complex biological systems. So there are a number of challenges related to 3D imaging, which include staining, imaging-seq structures, analysis of imaging stacks, and also resolving 3D biology and giving scientists the numbers. So we offer ImageXpress micro confocal imaging system with water immersion option, which allows very sharp and crisp resolution of these stacks in a volume of 3D tissues, and also custom model editor, which is extension of MetaXpress software, which allows customized analysis of complex biology, including counting and analysis objects within objects, and which could work with the system in 2D and also in 3D. So the typical workflow of 3D assay includes 2D, 2D culture, and also usually plating cells into low attachment plates, which allow formation of spheroids or organoids, which could be imaged using confocal imaging system. And then these stacks could be analyzed in 2D or 3D. And with 3D analysis, you can define volumes, count cells in a volume, and also identify distances between objects. In this example, you can see imaging of spheroid with these stacks. And this is 113 colon carcinoma cell lines and with 10x magnification. So you can analyze individual images or projection image, like in this case, this is maximum projection. And those cells stained with Hurst nuclear stain, also viability stains calcium AM and ethidium homodimer for dead cells. And then you proceed with analysis, which allows reconstitution of these structures and cells in a volume, and also provides a number which could be evaluated. So in this example, you can see projection image of control spheroid and also spheroids treated with different compounds. So you can see dramatic phenotypic changes in biology. So you can see this spheroid is shrinked, and in other example, you can see cell death or dissipation of spheroid, and you can analyze all of these changes and also describe it using software. So you can count cells within an object and also characterize the number and ratio of cells of different phenotypes, for example, live and dead cells. And also you can get morphological and intensity readouts. So then you can do similar analysis of each of these stacks, and then software converts this information in 3D space and gives you volumetric measurements about the objects. So here you can see two spheroids, one control and one treated with staurosporine, and you can see nuclei in red and cytoplasm in green, and viable cytoplasm which stained with calcium AM. You can see the changes in a number of cells, stained cells, and also the volume of cytoplasm. And you can measure all of these readouts, and you can provide the tables or spreadsheet of numerous readouts, which could be further analyzed. So we combine this imaging capacity with Puma system, Flowchips. So in our typical assay, we can place spheroids into the Flowchips and also pipette in media and compound dilutions, as well as staining reagents. Then you can incubate these samples with automated liquid exchange, and in a designed amount of time, typically it's 48 hours, you can take plate out and image it using XM confocal imaging system. So the processing of organoids goes on automatically, and the system allows efficient media exchange with different concentration of compounds, and also addition of staining solution and washing step at the end. Then, as a follow-up, we do imaging analysis, and those plates in a flow chip, very convenient for imaging and allow achieve very nice quality of images. Here you can see examples of 3D spheroids. So this is HeLa cells, and this is spheroid stained with nucleo apoptotic stain. So you can see sporadic apoptotic cells there. You can see staining with mitochondria, and you can see typical mitochondria stain pattern, which is around the nucleus, and you can see nuclear stain. So you can achieve very good resolution by using water immersion objective, which also works with flow chip system. So then you can follow up with image analysis and use custom model editor, which allow to identify spheroid as an object and also define individual cells within that object and do their characterization for different stains. And so as a result, you can see numbers which include numbers and content of different cell types, as well as their area, volumes, or intensities. In this example, you can see HepG2 cells stained with earth and ethidium homodimer, and analysis masks on a right, which show live cells in red and dead cells, which is ethidium homodimer positive in the center. So you can evaluate numbers or percentages of live and dead cells in treated or untreated samples. Here in this example, you can see HeLa cell spheroid and also HepG2 spheroids stained with different markers. In this example, you can see control spheroid, which has bright calcium AM stain and very few cells stained with ethidium homodimer. And you can see treated spheroid, which have majority of cells positive for ethidium homodimer. And here you can see analysis masks, which represent these changes. In this example, you can see a nuclear stain, ethidium homodimer, and apoptosis marker. And you can see also quantitation of different cell types using image analysis. So here you can see diagram presentation of different number of different cell types after treatment with various drugs. You can also see nice concentration response with different drugs. Here you can see this taurosporin response for HeLa cells or HepG2. So you can see dose response changes and dose response and increase of dead cells and decrease in live cells, which you can plot using a 4-parametric plot curve and define EC50s for live and dead cells. In addition, you can collect supernatants from these cultured cells and define secreted factors, in this case, VEGF. So for different compounds, which we have done treatment for, defined EC50s for live and dead cells. You can also go with 3D analysis, and in this example, you can see spheroids, 20x magnification stained with nuclear stain and calcium AM. And you can do 3D analysis, which allow to define number of live cells, number of dead cells, also volume of positive cytoplasm and average volume per cell of cytoplasm, as well as intensity. So all of those dependencies in this case follow a nice monotonous concentration response, and you can define EC50s for those readouts. So in another example, we use Puma system to do functional assays like calcium oscillations. So we've used microbrain structures from stemonics, and those organoids made from induced pluripotent cells, and they contain 3 main neural cell types. And they have spontaneous activities, which you can measure by calcium oscillation. So we add calcium dye to those organoids, and we observe changes in fluorescent intensities, which goes up and down. So here you can see a recording of fluorescent intensity for control organoid. Here's the one treated with gabapentin, which is agonist of inhibitory neurons. So you can see slowdown of oscillation and also increase of amplitude. Here you can see increase of amplitude and also increase of frequency after treatment with 4-aminopyridine, which is a stimulator. So those recordings were done by Flipper instrument. And also you can do imaging by recording on IXMC. And this is some examples of those recordings. So you can see fluctuation going up and down. And again, you can see difference of control sample in sample treated with aminopyridine. So here you can see kind of the image of all these organoids which line up inside the chip. And you can treat them with compound and study effect of compounds on calcium oscillation. So those measurements could be done by using Flipper instrument, and ScreenWork Pro 2 software allows characterization of those patterns, like to define amplitude, frequency, rise and decay time, and appearance of secondary peaks. So the advantage of Puma system in this case would be related to the fact that you can actually measure effect of compounds consequently with the same microtissues or organoid. So for example, you can treat spheroid with calcium dye and then add media or add compounds in increasing concentration, and you can take this plate in and out of Puma system and measure it on a Flipper instrument, and to do recording after different concentration or different times of treatment. Usually without Puma systems, this workflow would be extremely difficult because you cannot efficiently exchange the media with spheroids, because you kind of you can pipette it out accidentally, or it's very difficult to completely exchange the media. But by using Puma system, you're able to change the media completely and then to do dose response with the same spheroid, or study, for example, one compound and then wash it out and treat with different compound and do all these perturbations. So here you can see the example of spheroid, which was treated with increasing concentration of aminopyridine. And here you can see an organoid treated with gabapentin or azimilate. So in addition, you can actually follow up this viability stain, and you can define number of live and dead cells by using imaging as described in a previous experiment. So now we've recently started to work with tumoroids, and this is a very exciting project. So we do it in collaboration with Tulane University. And those tumoroids derived from primary breast cancer tumors. And so we use the system to simplify the workflow. So we were able to grow these organoids in suspension and then load onto the flow chip. and then we can treat them with compound and follow up with staining and imaging. So here you can see control organoids, tumoroids stained with calcium AM and etidium homodimer, and you can see decrease of cell viability with romidepsin, increasing concentration of romidepsin. And so then you can follow up with imaging and image analysis. Like in this example, you can see that software allowed detection of both organoids and detect number of positive or negative for calcium AM or etidium homodimer cells. So, and those numbers could be reported for live and dead cells for each of the spheroids. So those spheroids are mostly alive, and those which are going to have a lot of dead cells, you can see it with yellow stain, and you can report it with the numbers. So finally, I would like to describe the use of Puma system for simplifying organoid workflow with using Flowchips. So we've been working with lung organoids, which were derived from primary bronchial epithelial cells. The workflow includes expansion of cells in 2D and then culture in matrigel droplets. So with media exchange, and this process is pretty long and involves several weeks of culture and then maturation of those organoids. And then at the end, we would like to treat them with compound and see the effect. In our experiments, we wanted to see effect on cell viability. And typically, to work with organoid, it's really difficult to work with the system for screening assay, because you have to deal with these matrigel DOMs. So what we did, we actually diluted the matrigel and loaded those organoids onto the flow chip, and then loaded different compounds and study effect on cell viability. So this is the images which we get after staining with calcium AM and first. So you can see lung organoids. This is projection image. So you can see very nice images. And here, this is 3-color stain in addition with mitochondrial stain. And interestingly, so you can image those organoids using confocal system, actually pretty precise, and you can see the hollow structure and also complexity of their kind of morphology. In this example, so we treat organoids with ibrutinib for 48 hours and see effect on cell viability, which actually was much greater than with the same cells in 2D culture. So we follow up with etidium homodimer stain and calculated number of live and dead cells and define EC50s. So in this example, you can see how we use like live dead analysis, which allow definition of calcium positive cells in a control, and also this intermediate treatment with intermediate dose or high dose. So you can see decrease in the number and percentage of live cells and increase in number of dead cells. So in a summary, 3D microtissues important for current biology and understanding diseases, and they present number of essential technical challenges. And when you use microfluidics in combination with high content imaging, it simplifies greatly the workflow and streamline complex protocols. And we would like to acknowledge our colleagues from Protein Fluidics and Molecular Devices, and also our collaborators from Tulane University, Washington University in St. Louis, also UT Southwestern and Grainer Biobank. Thank you so much for your attention. Please proceed with the questions.
Very good talk, and we're already getting some good questions here. And so Before I end up reading some of those off so that the audience can hear, selfishly, I have a couple of my own. So we did talk a little bit about Matrigel. Let me know how you release organoids from Matrigel if you happen to be performing assays and growing them in Matrigel.
Oksana, do you want to guess that first?
Yes. So for this specific assay, so we just diluted Matrigel slightly and let them sediment and then load it onto the chip and proceed with treatment and also staining.
There's also a cell recovery solution that you can buy, which will essentially dissolve the Matrigel off. I think that's commonly used if you want to passage your organoids. And we've shown that if you use a cell recovery solution to dissolve the Matrigel, you can pipette out the organoids from your plate and transfer them into our Flowchips.
Great. When we talked a lot about imaging here, so another question is, since you've got fluidics and obviously COC structure that holds the organoid in place at the bottom of the well, are these designed for imaging, or are they suitable for standard plate readers as well?
They can be used on pretty much any downstream analysis system. So yes, plate readers, if you want to do fluorescence, we've done luminescence reads using the Cell Titer Glow assay kit. And these are clear bottom and open top. So, and then the wells, I think we mentioned, are on a 384 well spacing. So if you have a generic 384 well plate definition file in your system, you can typically use that to read our plates.
Great, thanks, Evan. Sounds like that was designed in at the very earliest stages. So
yeah, we want to make it work seamlessly with your laboratory equipment.
Well done, thank you. We got a question from Brandon. What is the diameter of the fluidics channels?
So the fluidics channels are on the order of 50 to 100 microns in size. The bottom aperture of the sample is 1.2 millimeters. And so you have a good clear view of your organoid in that.
Great. Some other questions coming in and some thank yous. Thank you, Adriana. Oksana, maybe you can tell us a little more about some of the image analysis that might be appropriate specifically for organoids or tumoroids.
So before I answer that, I've seen one question in question and answer, but for some reason my typed answer didn't go in. So the scientist asked how deeply we can image spheroids and organoids. Typically, it depends from what is the nature of those spheroids or organoids, and also what kind of imaging methods were used. So for something like stemonics organoids or tumor-made spheroids, which pretty solid, you can go into with 10x magnification, like up to 200 microns, and with some decent resolution that you can still see the structures and see different cells. But organoids, surprisingly, since they're more transparent and they have a cavity inside, you can actually image them much higher than that and much bigger structures. And about analysis, this is actually a very interesting question because different customers may use different imaging instrument for capturing complexity of biology. So you can do as simple as live dead imaging or cell count or cell scoring, characterization of cells for different markers, for example, to define apoptotic cells or cells positive for calcium AM or positive for mitochondria. But you can also do some more sophisticated analysis where you're able to count cells within other objects. So for this multiple spheroids and organoids, this is kind of very useful analysis which you can do using custom model editor. With this application, you can actually define spheroids or organoids first and then count different cells within them and also characterize those cells for live, dead, or positive or negative for different markers, as well as characterize, like estimate area or intensity of those. The most interesting example when you do 3D analysis. So with this application, you can not only count cells, but also define distance between cells, as well as volumes and intensity again, and you can define object within a larger object. So it's very interesting.
No, that's great. So if I'm hearing that right, you can actually have multiple spheroids per well so that you have even more data points per channel.
Yes, this is correct.
Very good. Scott is asking us, how inert is Matrigel? Can it affect the assay results or imaging?
So the Matrigel, I guess, is a lot of discussions about Matrigel. Its use, I wouldn't call it necessarily inert. It has a whole bunch of things in there. In terms of use in our system, the Matrigel itself, you need to remove that, because if you put pure Matrigel into our chips, you will clog up the flow chips. But as we mentioned, if you dissolve it off or use a cell recovery solution to remove the Matrigel, then once that's done, the organoids can go into our flow chips without any issues. And as far as imaging, Oksana, do you want to talk about imaging in Matrigel versus imaging in our flow chips?
So this is actually was a very good quality of imaging, and so I would say in a flow chip it was comparable than if you have in a standard wells, but since we use washed out Matrigel, it's we get quality of imaging even better.
Great. So we actually have a couple of questions that are showing up in the chat, so I don't want to ignore those. Lydia is asking us, how long does the instrument take to quantify infiltrating cells in a well? Are multiple wells quantified at once? Same question for imaging. I think that is an imaging question.
So imaging is typically done for one well at a time, and so it kind of depends on time of imaging and of acquisition and imaging and speed of analysis. So typically we image each well in about a second, depends on like how many wavelengths you're using and what is the exposure time. So it goes pretty fast.
Lydia also asked if you have to use Matrigel to hold the organoids in place, but Evan, it sounds like you've already addressed this, but I'll let you elaborate.
That's correct. So once the organoids are in that bottom protected chamber, they stay in place very nicely. We also have used a nano shuttle, which is a product of Griner Bio 1, where you can either use the little magnetic particles to help form your organoids, or you can coat the organoids and then use magnets to hold them in the bottom of the well. So we find that's useful if you have smaller organoids or things that might be a little squishy, that you can use this nano shuttle product to help place and hold them in position. It also centers them in the well, so it makes it a lot easier for the imaging systems to find them. You don't have to worry about them being in corners or moving around. So.
No, thank you. I'm a big fan of the nano shuttle, and actually Lydia's next question was, can we use spheres generating via nano shuttle?
Well, yes, you can.
Yes, you can. And then how large are the spheres? I think she was guessing 100 to 300 microns, but I guess that really speaks to the 1.2 millimeter diameter of the micro well and how deep you want to image, right?
Yes. So we've done spheres organoids up to sort of 700 or 800 microns, and then the islets I showed were typically around 100 microns or so. So you put multiple organoids in there. If you think about the number of data points you have, if you have one well with 4 or 5 organoids in there that you're processing, each organoid is an independent data point, so you can collect multiple data points on each one. with 32 sample wells in each plate.
Got it. Okay. And then Lydia was also asking, do you keep the plate on the nano shuttle plate? I'm wondering if she means by initial incubation, but I'll let you address that.
So we have a product we call the 3D mag, which puts little magnets underneath the plate when it's in our system. And so we sort of incorporate that into our Puma system as an accessory if you want to use NanoShuttle. We also have a loading tray that when you are loading the spheroids or organoids into our plate, it has little magnets which then help position it and center the organoids in the bottom of the well. So we've incorporated the magnets into our system, which is available as an option if you want to use an NanoShuttle.
Very cool. Well, I think our questions in chat are starting to slow down. I'll jump in if there's something else that comes up, but I want to thank everybody again for joining, and especially our distinguished speakers, Oksana, Sirenko, and Evan Cromwell. And if you have any parting words for the audience, please feel free.
Thank you so much for joining, and thank you so much for interesting questions. And some of those were very kind of provoking further thoughts and ideas for development. And a couple of people asked about internship opportunity. Of course, we open for intern here in the lab.
Yeah, and also thank you, Oksana, for all the great images and work you've done with our system here. And I encourage you to go visit our website if you want to find out more information on the Electrodevices website about the imaging. And my email is there, so you can always reach out to me directly. I'm always happy to answer questions or provide information on how you might be able to use our system for your organoid assays.
Thanks, Evan. Thanks, Oksana. And thank you all for listening. And with that, we'll go ahead and close the call.
Thank you.
Thank you.
Bye all.